Hello, everyone, and welcome to another episode of The Imposter. That's right, the podcast dedicated to making science more fun and enjoyable for you, the listening audience. So, because I've been in transit between countries and states within the United States and cities within those states, um, this episode is one from the archives that I've pulled, and it's a conversation that I recorded between me and a few of my friends from the master's course that we did at Plymouth University, and it's a casual conversation, but we talk about each other's projects and have some fun. So let's get right into it, folks.
We live in an age based on science and technology with formidable technological powers. And if we don't understand it, by we, I mean the general public, if it's something that, oh, I'm not good at that, I don't know anything about it, then who is making all the decisions about science and technology that are going to determine what kind of future our children live in?
We've really got to start at the earliest levels with having a broader view of what education really can and should be. Because I find that with the young people we have, we are able to motivate them.
Science is all around us. It's in us. Knowledge of science is power. It gives you an understanding of the forces of nature. It's not even about how much science you know. It's about how science literate you are.
Hello, everyone. Welcome to this episode of The Imposter. I am Amir Fogel, your lovely host, and today I'm joined by some very good friends of mine. We'll just go around the room because these are some ex-peers. No longer peers, but ex-peers. So we'll start with my left.
Hello, I'm Dean McKeown. I'm still your peer, by the way.
You're never my peer. All right, so we have Dean.
I'm Ro.
Hello, I'm Ryan.
And I'm Josh.
And the lot of us were on the MRes course at Plymouth University between 2014 and 2015. And this episode is just going to be kind of a casual conversation between some good minds. Kind of a mind meld, if you will. So I'm going to start us off, and I'd just like to maybe go around and see what everybody's project was, and or final dissertation, we should say. So we're going to switch it up and actually start with Joshua.
Oh, okay, yeah. So my MRes thesis was looking at bottlenose dolphins. I was trying to use mathematical models to predict where they might occur spatially in my two study areas, the Bay of Biscay and the English Channel. It was an interesting project for a few reasons, one of which was it hadn't been done before. It's especially difficult to do this kind of technique with a species that's wide-ranging, trying to pin them down to a certain area and then trying to define a decent area where you can protect them. And it was also interesting for me because I learned a lot of new techniques and I learned a lot of things I hadn't realised previously. Quickly to summary what I found, you can't do it. You can't. You basically. There's been a push recently for science to be published that finds kind of nothing, and this is interesting in that case because it hadn't been done before. I tried to do it and found it couldn't be done, at least the way I was doing it. It's so difficult to try and link the distribution of a species like the bottlenose dolphin to 4 or 5 disparate factors.
If you don't mind me asking, so then what are some solutions?
More data, more sightings data, more environmental data, maybe prey data, so where their prey naturally occurs. Basically just trying to plug that model, the model I was using, full of more. And then you might see a link, you might see something significant, but I didn't.
Do you think tagging data would be useful in that respect?
Tagging data would be extremely useful. In Europe, though, illegal possibly.
Really?
Really?
That's a memory I've got. I know in the USA...
That's going on record?
You can take skin biopsies with a modified rifle, but I don't know if you can tag a bottlenose dolphin, but I'm not certain. They're an Annex 2 species under EU law, so... I mean, I just feel like if you could, why haven't they done it yet?
Yeah, I mean,
that's a good point.
Interesting point.
Mm.
Yeah.
We'll have to look that one up.
There must be permissions you can get, to get around.
What if it's...
I don't know.
If it's already a wounded dolphin?
Sure.
if it's kind of limp
If it's
swimming,
looking for a place to die, then
So you can take tissue. You can literally take their skin, but
Yes.
not put a tag in them.
sure, yeah. Well, I mean, I'm just thinking back to all the papers I read about the species, and I can't remember one where they were tagged.
Doesn't David Sims tag?
Sharks.
Basking sharks.
Sharks.
So are they any different annex from dolphins?
I assume so.
They're not
They're not mammals,
for a
yeah.
start, so they don't have
I
feelings.
thought it was just vertebrates. I
Clearly.
thought it was vertebrates that were in their own special sort of protection category.
They are in terms of passing it through the ethics board. It's like any vertebrates or any
Cephalopods,
cephalopods
yeah.
come under something.
They're
called
honorary
ethics.
vertebrates, aren't they?
Yeah,
Cephalopods.
but it doesn't mean they're annexed species.
But mammals are even more protected. They're even more
Sure, and in the EU,
special.
I don't know if there's a marine mammal more protected than the bottlenose dolphin and the harbour porpoise. They're just,
Porpoise.
it's like, don't even touch them.
There's
Don't
purpose for
even
that
look at
porpoise.
them.
Don't even look at
Don't
them.
even
bottlenose
dream.
dolphins kill harbour porpoises quite
Yeah,
often.
sure,
It's
yeah.
quite an interesting thing.
Yeah, but
A
then
lot of people
foxes
don't
kill
know.
stoats.
No, I know. I'm not criticising the bottlenose dolphin. I just think it's an interesting.
Yeah.
interesting
Oh, yeah. They're
thing
very
that-
territorial.
Yeah.
Like, very
Well, who's
territorial.
teaming on?
Well, I don't
I'm
know.
on my
Dolphins,
side.
porpoises.
Do you know what I learned about otters recently? I know this is kind of a tangent, but everybody likes to think of otters as really cute animals, and they're really messed up. Like, there's some sexual
will
assault that
have.
goes on by dolphins, but otters not only perform lots of sexual assault on other otters, on dogs.
How
But-
can you define it as assault if you can't understand their language? Maybe there was
Actions.
consent.
I don't.
Well, exactly. I mean,
Okay, I'll tell you. I'll
it
tell you
sounds
how.
ridiculous
Because
but...
it's murder.
Oh.
I read a few anecdotal evidences. Of
Okay,
course, it's
yeah.
not empirical, but it's anecdotal. Where otters drowned dogs that were swimming and then proceeded to have sex with their bodies
Wow.
Hmm.
up to like a day or two after the bodies were actually dead. Like just floating around. They're like, yeah,
So
you're
you're
my sexual receptacle.
But Amir, they sleep holding hands.
Like, yes, and they also kill baby seals and have sex with their bodies.
Seals as well.
Seals
yeah, it's
as well.
unlikely they would
They-
have agreed to that.
Have you seen that? Like
Yeah.
Cross-culturally, it's hard to communicate.
I mean,
Yes, you can kill me. In return.
We're a liberal ocean
Yeah.
society.
Have you seen that story about the guy who was like working somewhere and it was some bird flew into the window and died, and then another bird came along. It was a duck, and it started having sex with the body for like quite a long time.
Wait, the
And
duck
he-
was having sex with the
with
dead
the
bird.
dead duck. And he just watched it for ages, and it got published as a paper. It's called like
What?
the necrophiliac gay duck. They're both male ducks as well.
It can't be called the neck food.
It is.
Nope.
Like,
That's what
the,
it-
Sure.
the title.
Not only that, it was in Nature.
Yeah, just
It was the front page of Nature.
just like wrote what was happening and made like photos and then wrote a little paper about it. It's out
You
there.
make it sound so quaint.
It's out there somewhere.
That's interesting in of itself that you can publish. You know,
Yeah.
you can.
You don't need big
Keep
data
notes.
sets. You just
Yeah,
need
yeah.
something horrific.
I wonder
Something
how
unique.
it is. I wonder because obviously we've only just talked about 2 species here, but what other species and or organisms have like
Or
having sex with dead bodies as something that's common
Well, humans
in their
definitely.
behavior? Like
birds, definitely.
birds do?
are
Is that like a
obsessed.
thing with birds? Necrophiliac
Yeah.
birds?
I feel like it is.
I mean, I believe.
How did we get onto necrophilia so quickly? I knew it would be about 15 minutes. I didn't think it would be this soon.
Well, listen, you can't go 9 minutes without mentioning. Let's
Should we realign our
Let's,
subject?
All right,
let's realize.
we'll realign.
I'm
So
gonna
that was
bring
that.
it back to that.
that was that. Josh, that was a
that?
very interesting project, even though you found no true
Mhm.
I did.
solution. Would
Sure,
you say?
I didn't find a solution to my problem, but I found a lot of useful things.
And sorry, last thing before we move on, but well, if anybody else wants to ask anything, go for it. But because, well, you said they were very protected bottlenose dolphins. Is that right?
Correct.
So how vital is it to find an MPA or include them in MPA planning then?
Well, the thing is, they're quite abundant, so I don't.
So
The
it's not-
rationale
you're basically
behind their
saying they're
protection
important.
is like, I think to prevent local extinctions. I think in the southwest of England, especially, there's a small amount, and it'd be cool to protect them because they're not established yet. And I think it was 1993 where they started being sighted again after an absence, like a large absence from this area. So to maybe try and protect them somehow. But I guess the real follow-on question is defining an arbitrary area of ocean and saying that's now protected for the bottlenose dolphin. Would it have any effect on their survival? What's driving their decline, if they are declining at all? Data is the issue.
What was their baseline you were going from? I mean, you just said that they had an absence in southwest England, is what you said?
Yeah, until the mid-90s.
Yeah. So what were they very prevalent there 100 years ago, or is there?
I don't know.
No data.
But they are now there, and they weren't.
Sure.
So should we try and protect them?
Interesting.
I don't know if there's baseline data.
Do you think that a big gap in researching how MPAs work and where we should define them and all that stuff? Do you think they're missing negative results?
Possibly.
That there's a bias towards positive results. So if you say, oh, this doesn't link to bottlenose dolphin distribution, that's really important to get published because someone else might do the same research and waste their time. Or you might have an MPA that's defined based on that and it has no relation to the bottlenose dolphin.
I think both of those points.
But do you think that there's, for this research area in particular, the bias towards discovery?
I think so because policy drivers, policy makers, they want answers, not further questions.
Yeah.
So you're saying we've done all this work, and really we need to do more. We need this. We need to ask this question. They don't want to know that. They want to say, look, we've got a European target by 2020 to get a cohesive network of MPAs. Just tell us somewhere we can make one.
Yeah.
And then even if you're screaming at them, this may have no effect.
Well, those people are clearly not biologists.
Well, no.
Yeah, but they want something to show on paper.
This is where science and policy don't meet.
Another question.
All right, wait. Save that because we're going to come back to that very, very topic itself. All right, Ryan, why don't you go on and tell us about your project dissertation?
I looked at the impacts of shipping noise on marine invertebrates such as shore crabs, green shore crabs, using the shore crabs just as a model species, looking at whether shipping noise does have a physiological impact on crabs, to which I found that I found some evidence to suggest that there was an impact. My methodologies did have flaws in, so I wasn't able to substantially prove the impacts. But there's certainly evidence for impacts, which has much larger consequences, not just for shore crabs, but for any species within areas that are subjected to shipping noise. So we've got most harbours and ports are in estuaries. Down in the southwest especially, the estuaries do have all the salmon running at certain times of the year. The bigger picture is, does the shipping noise have an impact on migrating fish up estuaries? That's originally what my study went to look at, but because of permissions and things, I had to stick with invertebrates. But I've shown some evidence, and there's other studies out there that have got some really good evidence for the impacts of shipping noise, negative impact on physiology, behavioural, and physical damages.
So, are you saying that we would find more deformed crabs inside harbours?
So it's not.
What
so much
are you trying to say, Ryan?
Crabs. So I looked at breathing rates in crabs. So what I found was that under shipping noise, crabs increase their breathing rates compared to no noise or ambient noise. So the negative impacts of that would be that they're spending more energy metabolizing, so breathing, rather than going out and foraging and mating. So there could be much larger effects on crab stocks per se if they're not expending their energy more wisely.
So they're hyperventilating and having panic attacks, which
are,
is essentially stopping them from foraging more.
That's extrapolating it. They're not so much as having panic attacks, but they're increasing their breathing rate. And the real question is, to what extent? I mean, we've got estuaries that have constant noises, ships going back and forth. how much is this impacting them during the daily routines?
If if their respiratory rate's getting high, that would indicate that there was a stress response, wouldn't it, to the shipping noise, and that stress response was increasing the metabolic demand of the crab. So
exactly. Yeah,
it looks like there's a physiological response to the crab that they then need to fuel,
right?
Which is going to mess
the
up
stress
their
response. Yeah, obviously is a negative impact, and they're not using their energy for better
as efficiently as it could be.
I wonder if they would acclimatise to a ship going past.
Well,
especially in a busy area.
there has been a study that looked at shore crabs and acclimatisation. It's also what I tried to look into. I'm not entirely convinced if there is. The other study showed that with a repeated playback of shipping noise, the crabs didn't their stress response wasn't reduced, so it was suggested that they reached sort of a maximum stress response when exposed to shipping noise. I actually looked at I actually played two different types of shipping noise to crabs. So shipping noise that was recorded from the estuary that I collected them in, and shipping noise that was recorded from a different estuary, and looked at different responses. And again, there was some evidence to suggest that there were differences in responses. So it could be the sound regime of the shipping noise itself. There may be differences there, but there needs to be a lot more studies into recording the sounds in estuaries.
Do you mean like could one ship make a sound that a crab can actually hear because of its hearing range and another not?
So crabs don't actually like.
It's a good question.
How do crabs hear sounds?
So crabs aren't actually able to hear sounds. What they feel is the vibration in the water column.
Are they good vibrations?
Depends.
Answer the question.
Well, clearly not.
Oh yeah, clearly.
So your study wasn't in situ, you had them in the laboratory?
I had them in the lab. We did look at...
You tried to do it in situ.
Because it came down to financial and logistical constraints. But that really, what happens with ex situ studies is we don't really know if we get the same results in the field. Because when you play back sounds in a lab 2 experiment, you've got a very different environment, so the sound itself behaves very differently. And as Dean was saying, how do crabs hear? Well, they detect the particle motion. Where I was recording sound, I was recording the sound pressure levels, and inferring this as being the same as particle motion. However, it's known that it's different. The trouble is, detecting particle motion is very difficult, so it's often overlooked.
So the crab's whole body picks up there's no like hairs or anything that are...
So they've got hairs on their legs and statocysts within, so on their nerve cells, they've got tiny little hairs that detect the motions of the
and then add the nerves into the brain. That's basically what we've got, isn't it?
Effectively.
How long is the lifespan of a shore crab? Are the species you were looking at?
And is that relevant?
That's what I was thinking.
All right, I'll take you through my train of thought. My train of thought is this.
Let it play on.
This is my train of thought. If you're looking to see if it's acclimatizing to shipping sounds, will it acclimatize? Does the time of exposure in the lifespan of that crab dictate if it will have an easier time or a harder time acclimatizing? Wait, and also in terms of future generations, does a first generation exposure make it that maybe its offspring would be more used to a louder environment?
There has been a study that shows that a larger crab actually has a greater or an increased stress response when exposed to shipping noise than a smaller crab. So you can infer that a larger crab being an older crab, but this goes against what you're saying. So these larger crabs
would have been
react more strongly to shipping noise than your younger crabs.
But as well, there's less.
So that's not evidence for acclimatization.
Yeah.
Did they perceive more particle motion, or was it not?
That's possibly the case.
Are they too slow and bigger to get into smaller places that smaller crabs could hide in, and know they had that option as well.
It's all questions we can't answer. But
Yeah,
it's all interesting.
It's
as
a
a
very
question
new
Mike
field,
can
and
answer.
like I said, the biggest problem is ex situ studies and the difference in how sound behaves in
Yeah.
the field. And
Yeah, fair enough.
Is there any evidence that under conditions where there's shipping noise, that crabs actually alter their behaviour? So is there any evidence that crabs will reduce their foraging behaviour when exposed to the shipping noise?
there is it. They do reduce their foraging under shipping noise, and another study which again had its flaws, which I wouldn't be in a position to say who it was, but there were its flaws within that study.
Throwing it down.
That creates a situation where it's a bit of a double-edged sword, where you've got the increased metabolic load on the crab as a result of the stress of the shipping noise, but then they're also going to be taking in less energy as their foraging.
The reduced foraging will be a factor of the increased stress response, so they're spending more time breathing, so their breathing rates are increased. So rather than foraging, they're actually sitting there, almost panicking under the shipping noise.
So the increased breathing rate was also associated with just staying still.
That's not something that I looked at, but
okay.
I would like to look at that, because they looked as though there was two different responses in my study. One was to increase breathing rate, and there were a lot of crabs that decreased their breathing rate. So potentially, there was
right.
So there was
But he was smaller than 0.05.
God no. There's the potential that there's two different behaviours going on. One would be to sit and wait out the perceived threat being the ship pass. The other one would be to find a hiding place. So the crabs were all kept, in my study, the crabs were all kept in the same chamber, which didn't have anywhere for them to go. So there could be differences in behaviour there, which need much further studies. I'm not able to infer anything from on that front, but
well, and also vibrations.
Potentially, there's two different behaviours that crabs can.
Yeah.
Can I ask a final question? I assume.
I've got
more. I've got more too, but go on. Oh.
Has a crab ever had sex with a dead animal?
Funny you mentioned that.
Well,
not only that,
Broadcast spawning.
we did go another 14 minutes without mentioning necrophilia. So that's pretty good.
There you go.
That's a record.
I would like to know, and I don't know if this is something you can ask, but it's more on the physiology of crabs. Do you think that crabs can get a head rush from hyperventilating? Like, do they extract the oxygen molecules when they're respirating very fast, and then they get dizzy, and then that dizziness throws them off guard and become more susceptible to predation?
Please tell us what happens.
Please tell us, Ryan.
What do you want me to say?
Do crabs get dizzy?
when they're respirating.
Do their inner ears kind of go all fudgy? You know what I'm
want to
saying?
see through the eye of a crab. Let us into that.
On a stone.
No.
Imagine your eyes on a
We
stone.
just cannot think like a crab like you can.
I've seen you get in there.
Uh,
Really just act a crab.
I think
I
it most...
haven't been with a crab for a long time now.
not since my college years.
I'd be crabby about it.
I think that most of the work on sound in the marine environment's focused on the things that obviously use sound, like dolphins and whales. Do you think
Don't sound so
are invertebrates-
disgusted.
Hey.
Things that actually understand and have evolved to perceive.
But
just
here's
ridiculous.
the point. Point I'm making is that everything in the sea does, but we don't really seem to look at it much. Like, no one would ever think, oh, crab must, click.
Yeah, and
make
Ryan
sounds.
didn't think that. He went to look at salmon and
Yeah,
they were
that's-
He was neglecting the crab.
No one likes the
But
crab.
I've seen things about how certain larvae of invertebrates will move towards the sound of a
Yeah,
coral reef being like all the clicks that fish make, all the weird sounds they make.
There is also evidence in some marine invertebrate larvae don't actually develop under white noise. So there is.
That's crazy.
So sound does have a physical effect,
but massively ignored, not studied. Wow.
Understudied, and it's the problems of.
Ro has something to say about this.
in aspects like reef noise and their role in invertebrate recruitment are studied, and it is important, especially in crustacean recruitment. So yeah, it's not an entirely neglected field. It might be a neglected field in terms of like the responses of adult organisms physiologically. I don't actually know that field particularly well, but yeah, it's not entirely ignored in marine biology.
And I would like to agree with that and add, well, semi-agree. For at least elasmobranchs, there's different studies and also not studies and just anecdotal evidence where like they take plastic water bottles and crinkle them up and it attracts great whites because they're like, oh, these are low vibrations. And it's just like what the vibrations of a squiggling fish would give off. But then, did anybody see that article that was like they played heavy metal, I think it was, or death metal, thrasher music? Like, yeah, and it attracted great whites because it was giving off the same vibrations.
So they try different genres?
Well, that's what I was going to say. Like, they should do the thing with babies and like neonatal care where they're like, classical music is supposed to stimulate the developing mind.
Classical music makes great whites go nuts. Feeding frenzies.
They make a mosh pit in the middle, like they're just bashing other sharks.
Start having sex with dead seals.
And only 4 minutes. Oh, all right. So, Ryan, that was enlightening. Thank you for sharing about the crabs and the noise. Mr. Rowe, why don't you go ahead and take us on to yours?
I looked at the effect of ocean acidification on invertebrate recruitment on coral reefs, and we basically showed that under conditions expected by 2100, invertebrate settlement and the abundance and presence of different important species on a coral reef are severely affected by the acidic conditions in a future ocean. Yeah, so
it's quite the mouthful.
Define important species.
Yeah. Why you got to be so discriminatory, huh?
Yeah. Well,
what are your keystone species?
This is a good point. Like, just to explain what you'd consider a keystone species or an important species, it's something that plays like a disproportionately large ecological role relative to the abundance of the organism in total. So a good example of that would be something like a grazing gastropod, like a grazing snail, because they'll control algal populations on the reef and stop coral being overgrown. They also are an important species for the feeding of higher organisms like fish. So if grazing invertebrates or grazing gastropods start disappearing from coral reefs, then there's going to be big shifts in habitat, for example, and fish populations and everything around them. The important species that we're considering here were grazing gastropods specifically, and a few crustacean species. So we really looked at amphipods, decapods, and copepods, which are all really important prey species for any zooplanktivorous organisms that live in the area. So we found pretty profound shifts in how they're being recruited to coral reefs. The grazing gastropods was probably our most significant finding, where we just found that any calcified organisms or any organisms that make shells like gastropods are really heavily reduced, and that's because their larval stages are really, really vulnerable, and then their post-settlement developmental stages are really vulnerable to the acidic conditions that we're going to be witnessing in a future high CO2 world. So that's obviously going to have big ecological impacts on coral reefs. And then secondarily, we just found a general trend of reduced abundance in crustaceans, which is likely to have knock-on effects for zooplanktivorous fish or any other zooplanktivores. It's quite an interesting project, and I think it's starting to look like it's providing evidence that there might be some feedback loops in habitat shifts on coral reefs. So these acidic conditions are quite damaging to coral reefs anyway, because corals are calcifying organisms, and this more acidification is going to start dissolving their skeletons and making them more expensive to form. But on top of that, with the reduction in grazing calcifiers like calcifying gastropods, the pressure that's put on coral reefs in terms of them being overgrown by algae is going to increase quite significantly. So that positive feedback is likely to just accelerate habitat shifts where coral reefs start disappearing and they get replaced with macroalgal dominant habitat types.
I'd like to zero in on one point, which is kind of what we were talking about with Ryan's, which is the difference between kind of the lab work and the in situ data and why it's important for your specific project.
Yeah, well, it was a big challenge with the whole field of ocean acidification, where lots of people have done studies in laboratories looking at the physiological responses of a single organism to acidified conditions, and they're finding all sorts of stuff. They're finding some organisms really resilient, some of them really suffer, and they also found quite significant differences even between congeneric species or closely related species. But one of the problems with all this physiological data coming out of laboratories is it's not possible to extrapolate that to a natural environment and draw any sort of conclusion or create any sort of model of what the world's going to look like in the future, or what these particular marine environments with these organisms within them are going to look like. And there's quite an influential paper that came out in 2015, which was basically just saying there's no point in doing work on organism responses to ocean acidification outside the context of their communities, because the shifts in ecological interactions between organisms who are under the effect of ocean acidification is actually going to determine who the winners are and who the losers are. Now, for this study that we did for my MRes thesis, we went to volcanic CO2 seep in Papua New Guinea, and
were you there personally?
Twisting the knife, bro.
Sorry.
I wasn't allowed to go collect that myself. Anyway, moving on.
Sorry, yes, go on, please.
So the professors went on a little trip to Papua New Guinea, and basically what we have over there is an area of volcanic activity where high CO2 gas, which is about 99% pure CO2, bubbles through the water column, and it creates an area of localized seawater acidification, which is comparable to the conditions we're expecting in 2100 under the RCP 8.5 scenario from the IPCC.
Is that a conservative prediction?
No, that's the worst case scenario.
Right.
Or I say worst case scenario, it's more like if we don't do something significant about our emissions imminently.
Is that scenario avoidable? Do you think we could?
Yeah, that scenario is avoidable, but it's it's
like if what lots of European countries have said now, like Germany said we'll be getting 80% of our electricity from renewables by 2050. Like if they deliver on that, is that?
So just as a tangent,
I take it that's within. That would be a better scenario than the scenario you've used in this.
It would be, but
okay.
Not necessarily by like an enormous amount.
Yeah.
So the IPCC when they did their project, they came up with 4 scenarios. One was the best case scenario. One was the if nothing changes scenario, and then there were 2 kind of middle bands.
Right.
So 8.5 was the worst, and there was 6, 4, and I think 2.5. And basically, at the moment, things look like they're going to fall somewhere between 8.5 and 6.5. So the 2 worst, the worst scenarios. But if everything comes through the Paris Agreement, then it might be towards the lower ends, which would be beneficial.
These CO2 seeps, how old are they?
On a scale of 1 to 10.
No, so they've been documented for at least 80 years, but it's almost certain that they're hundreds and hundreds of years old. We just don't have any records of that being the case.
How often are they found? Are there new ones found yearly?
No, as in, do new ones pop up in an area where they didn't previously exist,
or is the technology?
Or have they just been discovered more frequently?
So from the perspective of science working on the biology of these CO2 seeps, they're found. They'll probably find a new one every few years. Like they've found them now in the Azores. There's one in Papua New Guinea. I think there are 2 in Italy.
And are there any that weren't previously in an area which now are?
Not as far as I'm aware. I think all of the ones that people are working with at the moment have been established for a known, set period of time, and they haven't just kind of sprung up overnight and someone's pointed out to a scientist that there's CO2 seeping here.
Do you think that they could be old enough so that the communities that live around them have actually had a lot of time to acclimatize and adapt to the kind of CO2, and maybe might not represent the response that would happen from an anthropogenic CO2 introduction, which might be more rapid?
Interesting point. Interesting framing of the point as well.
Badly worded.
I think so. The assumption that we generally operate on when we're studying these seeps is that they're established communities. So that they have reached their, the ecological term for it would be an equilibrium community where it's no longer in its developmental stages and its successional stages. It's reached the point where it's going to be, and that's useful for our observations. In terms of how that's how we can use that to model future responses under essentially what we're saying is like that we're going to see a rapid change, whereas these things might have been exposed to it for hundreds of years. But in reality, these communities are like, although they've been exposed to it for hundreds of years, because they're so localized, they've not had time to genetically adapt necessarily. So the kelp species within a seeping region of localized acidification won't necessarily be genetically different from the kelp species outside. The only place we see that is in certain marine invertebrates, which have very, very, very small dispersal ranges. But yeah, it's a really interesting point. But basically, we treat them as equilibrium communities.
Can I add something onto that question, which is that with that in mind then, or maybe when it comes to the case of those very specific invertebrate communities, but when we're, you know how you have those coral nurseries now, and they're going out and they're planting, different corals to kind of rebuild it, but if, ocean acidification continues in the trajectory it's given right now, you can plant all the coral you want. If it's not resilient to kind of the acidity that's coming, essentially, then it doesn't matter. Is there a way that we can find different species, or in terms of, I guess, artificially reproducing more resilient species over time?
Yeah, I see
based on those natural kind of seeps and the communities that they form.
It's not necessarily relevant to the seeps, but just to address your first point in terms of like transplanting corals, there's massive variance in the resilience of different coral species and different genotypes within a coral species to acidic conditions. So like really the more coral that we're breeding, the more coral we're transplanting, the greater the likelihood that coral reefs as a habitat will persist rather than species-specific stuff.
Sure.
And then to address the second part of your point, yes, there's a assisted reproduction project in coral going on at the moment in Hawaii, which Ellie Vaughan, one of our course mates,
yes, good old Ellie.
And that was a grant won by a scientist at the University of Hawaii called Ruth Gates, and she, if I don't have some of the research that well, but I think what she's doing is identifying resilient genotypes in certain coral species. And then fragmenting them to breed them, and then to just keep selecting the most resilient coral species to temperature and acidification, and then to keep fragmenting them and breeding them. And then the idea of that probably would be to eventually start transplanting those hyper-resilient genotypes into natural populations, and then those genotypes would be at a competitive advantage to the less resilient genotypes, and hopefully that would keep some sort of balance in these communities as these conditions prevailed.
Is there any forethought to maintaining biodiversity in this case? So what I'm thinking is, in the area, when I was living in Mexico, in the Mesoamerican Barrier Reef, in the Tulum area, I should say, where I was, the coral there got horrible white band. White band, yeah. And, 65% of the coral was kind of decimated and gone within a year or two, something like that, because they were all the same species. They were all susceptible. So one example of why biodiversity is important is because if you have different species, they might have different resiliences to different diseases and so on and so forth. So if in the future you only have like 2 or 3 different species that are capable of kind of thriving on a reef, what does that mean for the biodiversity of that reef?
I mean, first of all, that's not the case. Like, it's not a case where there's only going to be, a couple of coral species that can survive. There's going to be a lot.
Yeah.
I don't want to just chuck out an arbitrary figure, but it's,
go on.
It's not like just a handful. There's going to be a lot of coral species, and then even amongst coral species that are susceptible, there's going to be genotypes which are not. And then I guess your point still applies in terms of like genotype diversity, and that would make a less resilient population to other stressors. But
another thing that's probably pretty unique about the coral is you've got the symbiont diversity to play around with. So that could be, you could actually be selecting for different algal symbionts that live in them, and that could be conferring the resilience on it. So you might not have to inbreed the coral.
For just the listeners' reference, that's the zooanthele?
Yeah, the zooxanthellae live inside the coral that photosynthesize and release sugars and amino acids, and then the coral provides the algae with a home and other nutrients that like work together in a symbiosis. Yeah. But you can select the different strains of the zooxanthellae, and that can create a more resilient coral. Yeah. But the coral hasn't necessarily changed itself.
Yeah, exactly. And it's just a case of making sure that these corals can accept the thermo-resilient zooxanthellae, which a lot of them can. Like, there's lots of research going on in the Red Sea about the relative effects of what zooxanthellae is colonising a coral to how resilient it's going to be to bleaching, which is really the main threat. It's a really promising area of research. It's a really interesting area of research, and there's a lot of like very, very good people working on it.
It's a great word, zooxanthellae. Can I also point out how frustrating the way they name coral diseases is? Like they're called like white band disease, white pox disease, white pox, white disease, black pox, black band disease. It just keeps going on and on. I'm like, Jesus Christ, how am I supposed to tell these apart?
Surely there must be a better way to do it. All right, well, that was very enlightening, Rohan. Thank you very much. Let's move on to our kelp individual.
Token Irishman.
Our token Irishman, our Bushmills advocate, our Guinness milk wiener.
This podcast is brought to you
Guinness
by
isn't
Guinness.
Is it not?
I don't eat meat, alright? I'm allowed to drink beer that goes through something.
That's how all dictators
It's about
start.
reduction. It's about reduction.
I'm providing jobs for Germany. It doesn't matter what else I'm doing.
Not sticking
Yes,
to
it
some
does.
ridiculous, principle of not touching an animal or looking at an animal. It's impossible.
What are you talking
Do you covet your neighbor's animal?
Yes, I do. I'm gonna get that rabbit.
It's mine now.
Filter all your beer through it.
I'm just pouring its fur and let it trickle through.
Homebrew.
Really adds a nice taste.
All right. Sorry, Josh.
I was going to say, Dean, you found out something that means nothing to no one, isn't it? That's a double negative. That means it means something to everyone.
Damn right.
Interesting. I like that. All right, Dean.
I prefer triple negatives.
Tell us about kelp.
So
Not too much.
my project was actually looking at when kelp have intercourse with another kelp that's dead.
Wait.
That was a good
That was 20 minutes. Yeah, 20 minutes from the last necrophiliac.
We're getting better, guys. The program's really helping us. No, I was looking. Basically, my project was to find an undiscovered group of viruses that infect kelp. Kelp are the biggest of the brine seaweeds, which you will see on any rocky shore. They are really interesting, despite the yawning motions I'm getting from some of my fellow podcasters. So
The term is peers.
Are they keystone species?
yes, they are.
Wow.
They are the. They
It's the first
are.
I've heard of that tonight.
They are the coral. They're the coral reefs of cold waters, basically.
I hear kelps are useless.
Except for cold water coral reefs, which are, of course, the coral reefs of cold waters.
But they're really deep.
Not all of them.
Black coral is a fairly
Temperate.
deep coral, isn't it?
Temperate cold waters.
They're the less interesting cousin of the coral reef.
The-
Massively.
Are cold water corals... Wait, sorry, sidebar. Cold water corals are mostly not the carbonate corals, is that right?
That's not right. Unfortunately.
Yes, right. You heard it here first.
Cold water corals grow quicker than cold water corals? That's not right again.
They generally have faces of the presidents.
Oh, I always knew Mount Rushmore was a coral.
It does
A
have
land
that
coral.
living.
I had my suspicions.
If you look close enough, their noses, polyps everywhere.
That's just because the
Skin condition.
forefathers were very zitty, you know. Bad.
Smallpox.
bad
The
skin.
smallpox looks like.
All right, sorry, I interrupted, Dean. Go on. And
So yeah, they're enormous seaweeds. They are used in everything. You've probably consumed them today without knowing. They use them in industrial processes. They're made to make dairy products, all processed foods. There's even some really wacky, weird uses, which I'll talk about later. But really important. They change the atmosphere by releasing iodine chemicals. They've been used to prevent deficiency diseases. Asian people eat millions of tons of them every year. It's probably why they outlive us.
If you were gonna go vegan, would you eat kelp?
I would. In fact, I actually do consume that. I'm vegan.
It took 48 minutes for you to tell us that you were vegan. That's a vegan record.
I didn't bring it up first. You brought it first.
That's true. Have you seen Cowspiracy?
So basically my project was looking for to see if they're infected by a virus, to see what that would mean for the impacts that it would have on them in the future, because they're basically depicted to, some kelp forests are predicted to disappear by 2100, the same year Rose said, under climate change scenarios, and then also there might be implications for the industries that rely on kelp. So
What are those industries? Sorry.
So alginates, they extract alginates from kelp. Alginates are these long chain polysaccharides that bind to metals, and they have really unique, useful properties. They increase the viscosity of things, so they bind stuff together. They're used to make gels, toothpaste, dairy products, asphalt, all kinds of things. And they're our main source of those. We don't really have much other alternatives. Some bacteria make them, but kelp we rely on a lot. And there's also the food industry. They're also added to cattle feeds and agricultural fertilizers. And the only known viruses in seaweeds are of ectocarpoids, which are very small filamentous species that we don't use in industries or eat. They're not in some ways they're not as important to humans. And the virus what it does is basically it hijacks the reproduction of the seaweed and makes the reproductive organs produce viruses instead of spores. And it has this sort of, it's called a latent infection. So it has this sort of weird, not typical infection cycle where it has a stable relationship with its host, and it doesn't kill the host. It just stays in the genome, and it becomes active at certain times, so it can synchronize its production of particles at the same time as the spores, and then that's how it maintains its relationship.
So it lies dormant.
It lies dormant.
But the kelp is still...
Kelp does what it does.
Yeah.
Well, they're very understudied. So when they've looked at some seaweeds with a virus, sometimes the virus does affect photosynthesis. In some species, it makes photosynthesis increase, and others it decreases. So really, nobody really has any idea what these viruses mean.
So, sorry to cut you off. I'm not sorry at all, actually. But what is the ramifications then?
So
Why are you? Why is this important to study?
So the same studies have also changed the temperature of seaweeds infected by these viruses, and what happens is some of them start producing more viruses, and it actually shuts down the reproduction, and others doesn't have that effect. So we don't know what will happen as the sea temperatures increase, whether these viruses could shut down the reproduction of basically all species of seaweeds. But for kelp, this would be especially important, and we're already seeing kelp forests starting to disappear. And one of the main things that happens when they start to disappear is the microscopic life cycle stage is the most sensitive to temperature, and that starts to produce fewer gametes. And you see certain species of kelp start to retreat and move towards the poles, whilst other species, which are able to produce gametes and spores at those higher temperatures, start to replace them. So we have a similar scenario in Plymouth, where we have a Japanese kelp and a Portuguese kelp, which are adapted to warmer waters, are actually starting to replace the native kelps. And there's not a lot. I think one of the main battle fronts between these species is when they're reproducing at the microscopic phase of their life cycle. So if you have a different virus in one species and another that behaves differently in different temperatures, let's say it shuts down the reproduction of a native kelp when the water temperature is raised, but in the invasive or kelp, it doesn't have that effect. That could be a major driving factor in the shifts related to climate change. But this is all my speculation so far, because absolutely nothing is known about them. So my whole project was to just find out whether kelp have a virus, and I was able to find that there is one in several species, and it looks like it does the same thing as in the ectocarpoid. So it looks like it takes over the reproductive organs in the microscopic life cycle stage, and then it fills them with virus particles, and then probably releases those to infect the spores.
It's my understanding, based on looking at those microscopic images that you did, when you say that it affects the reproductive system or process, correct me if I'm wrong, because I very well could be, but that it actually literally explodes?
Yeah, it fills to the point where the whole cell is just virus particles.
Yeah.
There's no organelles, there's no chloroplasts, there's nothing, it just goes.
And that's how you knew that it was a virus infected?
Well, we know because we use a stain that binds to DNA. And in a normal cell, you look at it, you can see, we use a special microscope that makes chlorophyll appear red, and DNA will appear blue with the stain. So you look at a normal cell, you've got a big old blue nucleus and red chloroplasts around the edge, everything looks normal and fine. Then you see these other cells that don't have any chloroplasts, there's no structure in them at all, it's just a massive blue, the cell wall's disrupted, it doesn't look at all like a normal algal cell. And the same cells are found in kelp and in the other seaweed that has a virus.
So getting down to it, the nitty-gritty of your project, sir, what would you say the cash money point is of your final dissertation?
I think the cash money point is showing that this virus has impacts on the photosynthesis, growth, and especially the production of the spores and the gametes. Because from visiting a kelp farm myself back in Stratford Lough.
Back in the old days.
Back in Stratford Lough. And from reading about all the industries that are set up mostly in Asia and all the wild harvesting practices done elsewhere in the world, it's completely dependent on you grow your kelp up and then you get the spores again and then you reseed those in a tank and then you grow them up and put them back in the water. It's completely dependent on getting those spores and gametes, and if you're getting less of those, that's going to make your whole industry really fragile, and you need to know what the virus means, especially if the waters of the world are getting warmer. I think that's probably the biggest cash money
Cash money takeaway point. Very interesting stuff.
All right, everybody. Thanks for tuning in to this week's episode. That is all we have time for. But don't forget to tune into next week when we continue our little mini-series on products that contain animal ingredients. And you don't want to miss next week. Trust me. A doozer. Very interesting stuff, in my humble opinion. And yeah, don't forget to like and share us on Facebook, SoundCloud, Twitter. We are also on iTunes now, though you have to search with my name, search with Amir Fogel on iTunes, because for some reason the Imposter doesn't show up. I don't know. I mean, I am narcissistic, but this just happens to be a fun coincidence. So there you go, deal with it. Anyway, all right, everybody, we will see you next week, and you have a good night. All righty.